this video we will discuss the effect of radiations on the Dsup transfected Dsup transfected mRNAs in the normal tissues. So in the previous video we have understood the expressions of these Dsups in vivo through their enhanced lipid nanoparticle systems. So in this case they will discuss the effect of radiations on those normal tissues that are transfected with these Dsup proteins or Dsup mRNAs. So here is the schematic. What they did is they inject this Dsup mRNAs into the tumor areas that can be seen here, and this is the schematic that they have developed, where the green region shows the Dsup transfected regions, and the regions that are surrounded by this whole circle is irradiated with the radiations. And they did their studies in the buccal tissues and the rectal tissues cancer sites. So in the buccal tissues we can see that there is a decrease in the gamma H2AX per nucleus signals, indicating that most of this gamma H2AX, most of these double-stranded DNAs are been covered by these Dsup proteins, whereas in the case of no treatment and GFP mRNAs transfections, we can see the high number of gamma H2AX per nucleus. And this panel C is the results for the buccal tissues, which is surrounding its cancer tumors, and panel D is the results for the rectal tissues which surrounds its tumor area. And here also we can see that Dsup transfected mRNAs tissue shows less gamma H2AX per nucleus compared to the normal case. And in the panel E shows the buccal tissues immunofluorescence images, where we can see that AF568 signals are high in the no treatment and also in the GFP mRNA transfected cells, whereas when they use the Dsup mRNAs, the AF568 signals goes down significantly. And similar results they have seen for this rectal tissues immunofluorescence image also, where we can see that AF568 signals are high for no treatment and GFP mRNAs transfected regions, whereas the Dsup mRNAs transfected regions shows a decrease in the AF568 signals. And now they wanted to check, or the important things that they wanted to see is that whether this radiation has any impact on the tumors. The radiation does not have any impact. The Dsup proteins strategy will not provide any positive outcome. And one of the examples that's shown here in the panel G, where the orthotopic tumors are injected on the orthotopic tumors is on the left hand side, and the area protected is on the right hand side. And in this case, when the radiation is injected, like at the -14 stage, at day -14 is considered at which the orthopedic tumors is injected, and at day 0 these lipid nanoparticles with Dsup proteins are injected. And then after 16 days they euthanize the animals and check the tumor volumes. And what they are able to see is that, and this is the final timeline at which they check the tumor volume. They also check the tumor volume intermediately by euthanizing the mouse. What they are able to see that in all the 3 cases the tumor volume does not get affected. So this indicates that the Dsup proteins does not have any direct roles on the tumors, but it's the radioactive particles or the radiotherapy strategies that plays a role towards this triggering apoptosis or killing the tumor cells through some strategies or through some signaling pathways such as triggering apoptosis. If the tumor cells itself are protected by the radioactive therapies, then these Dsup protein strategies will not be effective on those cancer treatment. So Dsup proteins, the strategies will be useful in the case where tumor cells are less protected by the radiotherapies. Then these proteins can be used to prevent the normal tissue surrounding this tumor area. So I hope you understood these proteins functions and its applications, in which case it can be used and in which case it can't be used. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.